Okay, I think I'm going to give it a little bit longer just because I don't know exactly what the delay is. All right, I think we should now be live and people should be able to hear me. So hi, welcome back to our spring seminar for the Living Earth Collaborative and also the Evolution, Ecology, and Population Biology seminar here at Washington University. This is our first talk featuring Dr. Christy Edwards out of the Missouri Botanical Garden. And yeah, I just want to say, we're excited to be hosting another virtual seminar that we've got a lot of great speakers lined up, and all of them this semester will be, again, virtual. And if the time doesn't always work for you, feel free to watch those seminars on the YouTube channel anytime you want. They're all there. We're collecting them, and it's really turned out to be a wonderful resource to have those on the YouTube channel. So let's go ahead and get our speaker ready to go. So let me give a quick introduction of Dr. Christy Edwards. So the story begins with Dr. Edwards was a young child. She would accompany her mother, who at the time was studying landscape architecture in the Front Range of Colorado. And so during these trips with her mother on her courses in landscape architecture, Dr. Edwards became very enamored with plant identification, right? And she's, this is a young Dr. Edwards, around the ages of maybe 7 or 8. This interest has only become more solidified during her productive career in plant genetics and conservation. So she began her professional plant career at UC Boulder, where not only did she focus on ecology and evolution for her bachelor's, but she also studied Spanish. She then went on to the University of Florida to further develop her phylogenetic and conservation genetic approaches. So she received her PhD there, and then she left the wet, balmy peninsula of Florida for the desert mountains of Wyoming, where she had her postdoctoral position at University of Wyoming in Laramie. After that, she then went on to be a research biologist with the U.S. Army Corps of Engineers. They have a research and development center in Mississippi. So this is where she kind of got a little lost and confused from her plant focus, and she dabbled in a little bit of zoology. She looked at bats and Asian carp. In fact, she was instrumental in developing new genetic tools to detect the presence of Asian carp in the Great Lakes. It's very cool. But then she traveled north up the Mississippi to where she is now in her current position as the Stephen, and I hope I say his name correctly, Camilla Brower Associate Scientist for Conservation Genetics at the Missouri Botanical Garden. She is one of the main reasons that MoBot conducts cutting-edge research in plant genetics on site. As far as I could tell, most of the excellent genetic research before Dr. Edwards joined the faculty at MoBot was off-site. So with this new position, MoBot is now able to do a lot of their genetic research on-site. But of course, Dr. Edwards isn't just focusing all of her energy on-site at MoBot. She's very much involved with the St. Louis ecological community. She's affiliated with SLU, WashU, UMSL, and the Whitney R. Harris World Ecology Center. She's very highly productive in publishing with numerous high-impact papers, ranging from molecular ecology, genetics, and other excellent journals. And another very impressive accomplishment of Dr. Edwards, this is very cool, and then I'll let her speak, is that she brought an extinct plant back to life when she rediscovered, and I'm not going to say this name right, but I think it's Dracaena umbricolifera, which is a tree species on the islands off the coast of East Africa. This tree had been previously thought to be extinct, and her team and herself, they went and actually found that it's still extant. So that's very exciting. All right, there's much more I could tell you about her, but I'm sure you'd rather hear it from her. So without further delay, please let me introduce Dr. Christy Edwards and her presentation on some of her recent work entitled Milkweeds, Seeds, and Genes, a Genetic Approach to Understanding Low Fecundity in a Threatened Milkweed. Take it away, Dr. Edwards.
Okay. All right. Thanks for the great introduction. That was great. Okay, so what I'm going to be talking about today is a work that we've done on a 3-year project that we just recently completed. I'm looking at the relationship between genetic diversity and fecundity in Mead's milkweed. Okay, so as most of you probably know, hold on, my cat. As most of you know, as biodiversity scientists, we generally recognize 3 fundamental levels of biodiversity, including ecosystem diversity, species diversity, and genetic diversity. And so as a conservation geneticist, I generally deal with genetic diversity. And this level of biodiversity is important because it produces the physical and functional differences among individuals in a population. And it promotes resilience and adaptive potential in populations. But I would argue that our knowledge of biodiversity is incomplete, and this is particularly the case for genetic diversity, largely because genetic diversity is not readily observable in populations. And the other thing is it's affected by things like the demographic history and life history traits of plants. And so these things can affect genetic diversity, but we can't always predict how they will affect genetic diversity. So we really have to go out and measure it. And I would argue that effective conservation, which is kind of my primary motivation for my research, it really requires that we have an accurate understanding of biodiversity. And so that's how I see the field of conservation genetics, which is essentially quantifying genetic diversity and ensuring that it's effectively conserved. So another thing that conservation geneticists do is we detect and prevent genetic erosion. So the causes of genetic erosion are things that are generally things that cause species to become endangered. So these things are things like habitat loss, population fragmentation, declines in population size. And generally what genetic erosion is just a gradual loss of genetic diversity over time due to things like genetic drift, genetic bottlenecks, decreased migration among populations. And then if you have decreased migration among populations, you can get increased inbreeding within populations. And the long-term effects can be pretty dramatic. So you can get the loss of adaptive ability and a decrease in fitness and reproductive success. So how does genetic erosion lower reproductive success? And so for many species, what it can do is it can cause a shortage of genetically suitable mates. And so in plants, this is not really a problem for most plants that are capable of, that are self-compatible. So they can self-fertilize or they can undergo inbreeding. And so those ones don't really suffer from a shortage of genetically suitable mates. But it's particularly a problem in plant species that are self-incompatible. And so there's quite a few species out there that have a genetic system of self-incompatibility. And so what happens is you'll have a pistil that has a specific genotype, and the pollen has to be a different genotype in order for reproduction to occur. So what this does is it essentially reduces inbreeding between close relatives, and it absolutely prevents self-fertilization from occurring. So because of that, in order to have successful reproduction occurring in a self-incompatible species, you have to have a genetically different individual. And another thing that's interesting about self-incompatible species is that many of them are clonal. This seems to be a kind of a correlated life history strategy. And it's thought that clonality essentially provides reproductive assurance vegetatively in species that may be pollen limited. So in a clonal species, what you have is essentially individuals that are connected underground via roots and rhizomes. And so what will happen is that several neighboring plants, they may look like independent plants, but they're all genetically identical to each other. So you have these are all forming one genet, and these are individual ramets. And in a self-incompatible species, the ramets are not cross-compatible with each other. So because of that, if you have a loss of genotypes in a self-incompatible species through genetic erosion, then this can cause the loss of cross-compatible mates, and that can limit reproductive success. So there's been quite a few studies that have looked at this in self-incompatible species. One notable one is Zizyphus Salada, which is a species that I actually work on. So this species actually has populations where every individual in the population is comprised of a single genet. So you might have 100 individuals in the population, but they're all connected underground via the roots. And 10 of the 15 extant populations in this species are completely uniclonal, and so they're 100% mate limited. It's also been found in other Asclepias species. This one is one that's related to the species that I'm going to be talking about today. And then finally, here's another example where they found an allele effect and a lack of cross-compatibility in ranunculus. And so in this title, they actually liken the kind of lack of cross-compatible mates as an allele effect. And indeed, it is an example. So for those of you, just as a reminder, for those of you who don't necessarily remember what an allele effect is, essentially an allele effect is an effect where you really need to have an aggregation of individuals in order to have population growth. And kind of in the reverse, small populations often have like negative population growth. So over here, this just shows kind of examples of an allele effect, a weak allele effect, and no allele effect, where you get the proliferation rate or population growth rate, and then this is the number of individuals. So when you have a strong allele effect, if you have a small population size, you can actually get a negative population growth rate. And then as you get to bigger population sizes, you'll get an increase in population growth rate. With a weak allele effect, you can have somewhat just kind of a stable population growth rate until you get to a specific population size. And then this is in contrast to species that have no allele effect, where you see that there's no relationship between population size and the population growth rate. And the way that you can test for an allele effect is that you can look at any component of fitness and see if there's a relationship between that and the number or density of conspecifics. So that's what we'll be looking at today. Okay, so there are actually quite a few ways that you can have an allele effect, so underlying causes of allele effects. So there can be things like ecological allele effects related to predation. So for example, there's been studies that have shown that more dense populations experience lower predation rates. So there was a paper looking at seed predators, and more dense populations had lower predation pressure on their seeds. Probably the most common type of allele effect that's found is allele effects related to reproduction. So there can be two ways this may happen. One could be that you get reduced pollinator attraction in small population sizes, so you get relatively low reproduction rates. The other way is essentially pollen limitation, so just a reduced probability in small populations that pollinators will actually get the pollen to a conspecific. And then finally, there's genetic allele effects. So inbreeding depression is now widely recognized as a genetic allele effect. And then the one that we're going to be looking at today is an allele effect related to the loss of genetically compatible mates. Okay, so to test for an allele effect and to support the hypothesis of an allele effect, there's a few things that you need to have documented in the population. So generally, the first thing you need to have essentially a relationship between population size and genetic structure, where as you get a decrease in population size, you get a decrease in genetic diversity. The second thing you need to have is essentially as you get a decrease in genetic diversity, you need to get a decrease in the component in fitness. And then when you have both of these conditions satisfied, then if you find a positive relationship between fitness and population size, then that is evidence for a genetic allele effect. So that's what we're going to be looking for today. Okay, and so the species that we were looking for a genetic allele effect in is Mies milkweed, Asclepias medii. This is a long-lived perennial plant. It's clonal and self-incompatible. It has bee pollination, and then it has wind dispersed seeds. So the seed pods, it produces seed pods that contain, up to hundreds of small wind pollinated seeds. And then the species occupies mature untilled tallgrass prairie, and then also rhyolite glades down in southeastern Missouri. So because the species is a tallgrass prairie endemic, it's facing declines. So this is a map just showing the original extent of the tallgrass prairie, which is in light green. And then it also shows the remnant tallgrass prairie. And overall estimates of tallgrass prairie loss generally show that, but basically between 1% and 5% of tallgrass prairie remains. So this is a habitat that's just experienced massive losses. And because of that, tallgrass prairie is really in these tiny little fragments, and there's just very little left. So because of that, Mead's milkweed has been declining for decades. And so it used to occupy 5 states across the Midwest. But the species has been extirpated from both Wisconsin and Indiana. It has only a few small populations in Iowa and Illinois that are just barely hanging on. And then the majority of the populations are found in eastern Kansas, eastern Kansas and western Missouri. And then you have a few populations that are here down in southeastern Missouri, and those are the glade populations. And anyhow, so because the species is declining, it's listed as threatened, federally threatened. So in addition to the declines that it's faced because of habitat loss, another issue with the species is it has really low fecundity. So one of the reasons why it has low fecundity is because of the way tallgrass prairie habitats are managed. Many of the habitats are managed by haying. And so the issue is that haying usually occurs in June, May, June. And Mead's milkweed essentially starts flowering in May, but it doesn't actually mature its fruits until September. So if a population is hayed, it cuts the fruits off before the species can reproduce. So essentially, any sites that are hayed essentially have zero fecundity. But the other issue is that even in populations that aren't hayed, you get relatively low fecundity. So in many populations, you may have many, many flowering stems, but they might not produce any seed pods. However, there is some variation among populations in their reproductive success. So some populations kind of reliably produce fruits, and then the vast majority of them produce 0 or maybe a single fruit each year. So as all of you are probably aware, it's really important for a species to reproduce sexually in order to maintain it as a viable species. And so there's been a lot of interest in trying to understand why this species has such low fecundity. So there has been a few previous studies on fecundity, and the first 2 were conducted in the early 2000s and looked at some ecological factors on the fecundity of Mead's milkweed. So these 2 studies looked at a single large population, and then essentially what they did is they looked at the effect of a variety of factors on individual level fecundity. So these factors included things like management, herbivory, pollen limitation, and abiotic conditions. And so in this study, what they found was that in years where there was fire and greater rainfall in a previous year, that increased fruit production. So this is important because essentially what this shows is that fire kind of stimulates reproduction to occur, or it stimulates flowering. However, because this study essentially just looked at a single large population, which is actually has relatively high fecundity rates, they couldn't figure out, they had no way to understand why populations had some populations have 0 fecundity. They just weren't able to test it. And then there have been a couple of previous genetic studies. So the first was published in 1998. And this study looked at range-wide patterns of genetic diversity and the effects of management on genetic diversity. And in this study, they found that 74% of the genetic variation was maintained within populations. So essentially, they found that relatively high genetic diversity in populations. And the other thing that they found was that burned populations had more genets, so a greater number of genotypes, and a lower clonality than hayed populations. So these two populations here were burned, and these populations here were mowed or hayed. And this line here shows the number of genotypes. So there's just more genotypes in burned populations than in mowed populations. And then the bars show the clonality, and the burned populations had lower clonality. Another conclusion of their work was that generally they thought that clonal spread in the species and the attrition of genotypes may limit reproduction. So essentially they kind of set forth this hypothesis that reproduction may be limited by mate limitation. Or a genetic allele effect. However, because the majority of the populations that they studied were hayed, they weren't able to measure fecundity. So they were never actually able to test for a genetic allele effect. And then another study was published in early 2000s. And this one looked at the clonal structure of populations in Mead's milkweed. And generally the conclusions in this one were the same. Overall, they found, once again, really high genetic variation within populations. And then they also found fewer genets and more stems per genet in mowed populations. So this is an estimate of clonality, and burned populations had much lower clonality than mowed populations. They also suggested in this study that the attrition of genotypes may limit reproduction, but they thought that this may occur only in mowed populations. But once again, they kind of hypothesized that low fecundity may be the result of a genetic allele effect. However, once again, they studied a bunch of mowed populations, so they were unable to measure fecundity, and so they weren't able to look at the relationship between genetic diversity and fecundity. But so those two previous studies were really kind of instrumental in kind of infusing into the literature this idea that low fecundity in Mead's milkweed was likely due to a genetic allele effect. So the idea kind of permeated the literature, and it's been kind of suggested in almost every paper I've read about Mead's milkweed since those two original papers. So the idea is that fragmentation, reductions in population size, and genetic erosion have resulted in populations that contain only a couple of closely related genotypes. And this has essentially caused the lack of cross-compatible mates, and this has reduced fecundity in the species. And so that's really what we set out to test here. And so because this has been kind of a predominant idea that's been kind of voiced in the literature repeatedly, there's actually been quite a few management activities that have been conducted in order to address the issues of mate availability and to increase reproduction. So for example, the Missouri Department of Conservation has conducted a really extensive augmentation program where they've transplanted individuals across prairies all over Missouri with the goal of increasing mate availability and reproduction rates. And so they've been doing these for 10 years now. And so the issue is that we don't actually have direct evidence that fecundity is related to low genetic diversity in populations. So, the question really has been, are these efforts really going to increase fecundity in populations? Is it really appropriate to be transplanting individuals among populations or between major geographic regions? And, is could this actually be a problem by causing outbreeding depression, by transplanting populations into sites where they're not adapted? And so basically, we set out to test whether a genetic allele effect is responsible for low fecundity in populations of this species. So essentially what we did is we sampled quite a few populations and tested whether fitness in populations was associated with genetic diversity, genotypic richness, relatedness, and then also look to see if we could use the results to try to devise a management plan to increase fecundity in populations of the species. Okay, so here is a list of the study populations that we chose for this project. And basically the main criteria for inclusion in the study was that we needed to have a reasonable population size, but also we needed to make sure that the population wouldn't be burned or hayed during the study, because we needed to be able to collect fruits and count seeds to have an estimate of fecundity. So we ended up with a list of 12 sites that were spread across eastern Kansas, then kind of western Missouri, and then we had a couple populations in the Rhyolite Glades and the St. Francis Mountains. Okay, so for each site, essentially what we did when we first got there is we essentially combed the prairie to find every single stem of Mead's milkweed that we possibly could. Then what we did is we got a GPS point for every stem, and we put a tag in the ground. And then for each individual, we collected leaf tissue for DNA analysis. And then throughout the growing season, we revisited the populations 3 to 4 times, and we collected data about flowering, fruiting, and then we bagged the seed pods because the seeds shatter. And then we went back and collected seeds so that we could get estimates of fecundity. Okay, so then we took the leaf tissue back to the lab and genotyped every individual in all of the populations. So we ended up with a data set just under 1,200 individuals. And we genotyped them with 18 microsatellites. Okay, so then once we had the microsatellite data, then we did an analysis of clonality. So what we did is we assigned ramets to genets, and then we mapped the genetically distinct individuals. So we ended up with graphs that look like this, where each one of these dots represents a genet, and the size of the dot represents the number of individual stems that comprise the genet. Okay, so this gave us a number of genets in each population. And then it also allowed us to use the flowering data to get a number of distinct genets that were flowering in each population. So this is essentially the flowering population that would have been available for reproduction in each site. And then we also took the data and essentially did genetic diversity estimates. So we had 2 data sets that we used. So the first contained all the ramets, so just the raw microsatellite data. And then the other data set was essentially a data set composed of a single individual per genet, so with all replicate ramets removed. And then we just analyzed every genetic diversity metric we could think of. So things like number of alleles, allelic richness, private alleles, observed and expected heterozygosity, inbreeding coefficients, mean relatedness. And then we also did an analysis of genetic structure to understand, the relatedness of individuals within and among populations. Okay, so then we took all of these variables that we had and we analyzed correlations among them. So the predictor variables included a range of demographic variables, including things like the number of individuals or the number of ramets in the population, the number of genets in the population, the number of flowering ramets, the number of flowering genets, the density of flowering individuals. And we also had estimates of genetic diversity, including observed and expected heterozygosity, alleles, all of the genetic metrics. And then essentially we just went through it and looked for correlations between these factors and the estimates of fecundity. So our estimates of fecundity included the number of fruits that were produced per population, the number of clones producing fruits, and the total seed set per population. And so actually from here on out, these three estimates of fecundity were very highly correlated. So I'm just going to show everything using the number of mature fruits. Okay, so here is just a simple table showing the fecundity estimates of the populations. So here's our 12 sites, and these are the number of stems that we collected or surveyed and analyzed fecundity in each site. And then this is the number of ramets that flowered in the population. And so what you can see is that overall that out of just under 1,200 individuals, 273 of them flowered. So about 24% flowered. But what we found is that we had really, really low fecundity overall. So overall, only 4% of the stems that we included in our study actually set mature fruit. And as had been reported to us previously, we did observe a really big, large variation in the number of fruits that were produced across populations. So half of the populations produced 0 fruits at all. Several of them produced 1 or 2 fruits. And then there was 2 populations that were responsible for the majority of fruits produced in the study. So one site, Lexington Lake, produced 27 fruits. And then another one, Weimer Hill, produced 11. So as we had observed previously, there's a wide variety in fecundity, with a few populations producing large amounts of fruits and a large number of populations producing 0 or only 1 or 2 fruits. So in looking at the genetic diversity, so these are just estimates of genetic diversity across populations. And one of the first things I want to point out is, so here's the number of ramets that we sampled, and then here's the number of genets that we found in each population. So one of the things I think is the most important takeaway here is that even when you had a very small population, so for example, this site had 8 ramets, and we actually had 4 genets in that population. So 4 individuals that were capable of potentially reproducing. And in many sites, we had a really high number of genets. So this one had 70 genets. So there's actually relatively high genotypic diversity in the populations, which is somewhat surprising, given these kind of predictions that low genotypic diversity was occurring in the species. And not only that, we also saw relatively high allelic diversity, high numbers of private alleles, and relatively high estimates of heterozygosity. And then if you look over here in the right column, you have the inbreeding coefficient. And the inbreeding coefficient ranges between +1 and -1, with values around 0 indicating essentially just outcrossing populations. And what we found is that we didn't see really strong evidence of inbreeding, which is kind of what you would expect in a self-incompatible species. But overall, in some, we just found relatively high genotypic richness, high genetic diversity, and low inbreeding in these populations, which was actually really surprising. Okay, so going back to our test for genetic allele effects, the first kind of condition that needs to be satisfied is that we need to have a relationship between population size and genetic diversity. And we did observe that there was a significant correlation between the 2 variables. So populations that were smaller generally had lower observed heterozygosity, and and this is average number of alleles. But actually, this is not a surprise. This is actually a really common phenomenon where you see a positive relationship between genetic diversity and population size. But what I wanted to show this because I wanted to mention that we did see the first kind of condition supported. So, it was a two-step process to support a genetic allele effect. And we did see that the first condition was met here. Okay, so let's look at the genetic structure. So this is the results of our analysis of molecular variants. And what this does is it looks at the hierarchical partitioning of genetic variation within and among individuals and populations. And what we saw here was that overall, we had the vast majority of our genetic variation found within individuals. And we also saw that there was almost no among population genetic variation. So essentially what that shows is there's really no genetic structure in the species. So we don't see that like certain populations are genetically divergent from others. And this is true regardless of whether we use the data from all of the ramets or the data from all of the genets, which was also somewhat surprising. I thought there would be some sort of genetic structure. And another way to show this is a plot from a program called Structure. And essentially what this program tries to do is it tries to group individuals into populations and maximize Hardy-Weinberg equilibrium. And these plots show the results of the structure analysis. So each one of these blocks here represents a population. And each one of these lines represents an individual. And the colors represent the genetic groups that the program has placed individuals into. So essentially when you see, for example, an orange individual, essentially what that shows is that individual is more closely related to other individuals that are predominantly orange than the ones that are blue. So generally when you see strong genetic structure, you might see a pattern like this, where a single population is all composed of a single genetic group. And a lot of times when we look at endangered species, we'll see that each population contains a different genetic group. And what that means is that the populations that are within each population are more closely related to each other than they are to individuals in other populations. But in the case of Mead's milkweed, what we see is that individuals in one population are more closely related individuals that could be on the other side of the range of the species. So what this shows is that there's no genetic structure. Essentially, individuals are more closely related to individuals in other populations than they are to ones in the same population. And we see this regardless of the number of genetic groups that we use. We do see a slight effect of site, in that the populations over here are the ones that occupy the rhyolite glades. So we do see a slight effect of this kind of big geographic range disjunction. But other than that, we see basically no genetic structure. So because we see high genetic diversity and no genetic structure, I was actually not particularly surprised when we looked at the correlation between genetic diversity and fecundity, because essentially what we find is that there's no significant correlation between those two factors. So, like I said, the first condition of testing for a genetic allele effect was met, but the second condition, where we see that genetic diversity and a component of fitness should be correlated, we don't see that here. And in fact, none of the genetic parameters that we analyzed in the study were significantly correlated with fecundity. And this is actually backed up by some parentage analysis work that we did. So we collected seeds in all these populations, and we germinated them, and we collected leaf tissue. And since we knew the genotype of the mom that they were collected from, we used this analysis to essentially conduct parentage analysis to try to identify the pollen donor. And what we found is that the pollen donor of the seeds that were produced in the study was generally located within a half a kilometer. So what this suggests to me is that there is sufficient genetic diversity present within sites for reproduction. So something else is going on. So once again, just to summarize, so here's the evidence needed to test for an allele effect. genetic allele effect. And as we said, we saw that populations that had smaller size generally had lower genetic diversity. But what we did not find is a relationship between the component of fitness and genetic diversity. So essentially, we do not have evidence for a genetic allele effect. So in general, what we find is that populations are genetically diverse and they contain multiple unrelated genotypes. And we think that the reason why is because tallgrass prairie used to be this like long, uninterrupted, continuous habitat that occurred kind of throughout the Midwest. And we think, you'd have these big populations that would reproduce, and they had these wind dispersed seeds, and it would just move genetic material all over the tallgrass prairie. And this probably happened up until maybe 100 years ago. And Mead's milkweed plants are long-lived plants. They could probably live decades, 50 to 100 years. And so we think what we see is this kind of signature of this past panmixia that occurred 50 to 100 years ago. So we do not find support for a genetic allele effect contributing to low fecundity in the species. So what is it then? So the two variables that were most strongly correlated with fitness in the population, the first is the number of genetically different individuals that are flowering. But actually the most strong correlation that we observed in the study was simply a really strong correlation between the number of flowering individuals and the number of fruits that were produced in the population. And the thing that I think is the most interesting is it looks like there could be a threshold effect where you see that populations that have under 40 flowering ramets in the population don't have good reproductive success. And then once you hit somewhere between 50 and 60 flowering ramets in the population, it seems like you may get a dramatic increase in reproduction. Because fitness is related to flowering, we think that the allele effect that we observe in the species is actually related to pollination, which is actually the most common type of allele effect that you see. So what we think is that essentially that a certain number of flowering individuals must be reached in order for pollination to occur. And this has actually been found in other milkweed species. And there's been quite a few studies that have shown that milkweeds can be pollen limited. There are a couple of different underlying causes that could contribute to this allele effect. The first is pollinator limitation. In this situation, pollinators just might not be attracted to smaller populations. There was a pollination study that was conducted, or it was published in 2017 by some of my colleagues at SLU. And essentially what they found is that there are there's three taxa of large bees that are the pollinators of the species. And so it's certainly possible that that pollinators just simply aren't attracted to smaller populations of Mead's milkweed. And so this is something that we'd like to test. So we've kind of thought of an experiment that we'd like to do in the future to test pollinator limitation, which essentially would involve comparing pollinator visitation rates, the pollen area removal rate, so the pollen removal, and fecundity across populations that vary in numbers of flowering ramets, with the expectation that if these populations are pollinator limited, then small populations will show lower pollinator visitation rates and pollen area removal rates and lower fecundity. The other option is pollen limitation, which is essentially just kind of a phenomenon where there's just a lower likelihood that pollen will be transferred to a conspecific in a smaller population. Just harder for pollinators to get the pollen where it needs to go in a population where there's not a lot of other plants. And this, I think, is probably more likely to be the case because of the pollination biology of milkweeds. So I don't know if all of you are familiar with this, but essentially the pollen in milkweeds is packaged into these sacs called pollinia. And essentially this whole structure called a pollinarium is how the pollen is transported. And so this will get stuck to an insect. And then in order for successful pollination to occur, what will happen is this pollinia has to be inserted into the stigmatic slit here in the corona of a milkweed. So it's actually a relatively complicated process in order for pollination to occur in milkweeds. And so to test for this, we'd like to do an experiment where we compare fecundity in open pollinated and hand pollination treatments across populations with varying numbers of flowering ramets. And we would expect that in small populations, if they're pollen limited, that you'd see a big discrepancy in open pollination versus hand pollination, whereas in a larger population, you would see a relatively small discrepancy between open pollination and hand pollination. So that's something that we would like to do in the future as well. And if there is a issue with pollen limitation, then because the species is self-incompatible, that can actually kind of contribute to pollen limitation. Okay, so let's get into management implications. So in general, what we need to do is to reach a critical threshold of individuals flowering at once. And so there's quite a few populations in beads milkweed that don't even contain 50 stems, let alone 50 flowering stems. So in these populations, we need to do some augmentations to increase population size. The nice thing about this is within regions, there should be no need to worry about outbreeding depression. And so it's good that the MDC transplantation program was conducted because it actually is probably doing something to increase fecundity in the species. And indeed, we've actually took some of the seedlings that we germinated and did augmentation. This is a site that was burned. It's called the Barrier Conservation Area. And we essentially did an augmentation to increase population size in this population. And then second, in terms of management application or implications, I think the other thing that we should be thinking about is how we're burning. So if you think about kind of the way tallgrass prairie once was, there's probably large fires that burn large tracts of prairie at once. And so as we've seen previously, fire kind of resets flowering in the species, so it kind of synchronizes it. And so all the plants probably flowered concurrently and then causes big bursts of reproduction to occur. But currently, the populations are both spatially fragmented, but they're also managed by a whole bunch of different entities. So each small patch of prairie is managed by a different entity, and they're managed differently. So they may be burned in different years. And so we never get kind of a burst of post-fire flowering all at once. I think in order to increase the threshold of individuals flowering, we need to manage with fire in ways that will promote concurrent flowering. So for example, you could burn as much of a population of beads milkweed as possible in the same year. If there's multiple populations that are being managed by different entities or management agencies, then they could coordinate together to burn the populations all at once. So you get this kind of burst of flowering in response in the following year. And then finally, something to think about that we're kind of interested in investigating in the future. So the site that had the greatest fecundity, interestingly, was Hage for 30 years. And then it was burned in the dormant season before the study. And this population just had a big burst of stems and a big burst of flowering, and then produced 27 fruits. And so this kind of got us thinking about, well, what about the management of this population really caused this burst? So there's two things that we were wondering about. So one is whether if we did some increased haying, if it would increase clonality, which would help to kind of produce the number of stems in a population, so that if you burned after that, then maybe you would get an increase in population size, an increase in flowering stems. The other thing that we're wondering about is whether the long fire return interval in this site was the reason. So for example, if you have a long fire return interval, is it allow the plants to kind of build up resources in their rhizomes so that when you actually burn, you get a big burst of flowering. So these are two things that we'd like to investigate, both whether haying increases flowering after haying and then a burn increases flowering, or whether if we have a longer fire return interval, if that would increase flowering. Okay. So in conclusion, I think this study is interesting because there's just been this really long-standing kind of assumption in the literature that the reason for the low fecundity in the species is just because of low genotypic richness in populations. But it turns out you really need a study where you look at genetics and demographics and fecundity across multiple populations in order to test that. So this study, I think, was unique in that it kind of provided unparalleled insights into the population. And I think it's interesting, a genetic study really is necessary to rule out genetic causes. And I think it also is good because it really does provide a kind of a framework for the management of the species to hopefully improve fecundity in the future. Okay, so with that, I'd like to thank some of the people that work closely with me on this project. So Matthew Albrecht, my former technician, Shannon Scara, Noah Dell, who is a technician with Matthew. This is Lance Jesse, who knew all of these populations of Mead's milkweed and tromped around the prairie. He's like over 70 years old, and we were like struggling to keep up with him in the field. And then also my colleague, Melissa Brigler, who has worked closely with me on funding for this project. All right, so thank you.
Excellent. Thank you, Dr. Edwards. That was absolutely fantastic. What a great semester, or what a great seminar to start the semester off with. This is just a reminder to all of you that have watched. Please leave your questions in the chat on the YouTube channel. We do have a couple right off the bat, so I will pause for a second to let Dr. Edwards get a drink. There we go. And
I'm good, thanks.
take a breath. And let's start with, so I believe the first question comes from John Birmingham Jr. And he asks, is there evidence for selection for hypervariability at the loci that control self-incompatibility?
So unfortunately, we don't. So we do not have the S locus identified in Asclepius. So it has not been characterized. So unfortunately, we were unable to genotype the self-incompatibility locus directly, unfortunately. But that was actually kind of why we did these studies of parentage analysis, so that we can kind of get an idea of who was cross-compatible in the population. So unfortunately, we don't know the answer to that question.
Hey, thank you for a great talk. I'm going to ask a question in the chat from Brog Mashman. Do you know if the lack of genetic structure you find in Mead's milkweed population is found in other prairie remnant species? Or maybe the structure is unique because other species are short-lived?
Yeah, so this is the first study I actually ever worked on in a prairie species. Generally, a lot of the endangered species that are found around here are glade species, which have these kind of naturally fragmented habitats. And in those species, you really see very strong patterns of genetic structure. This is the first a really long-lived prairie species that I've worked on. And I think that in species that have shorter lifespans, I would expect that you would see like stronger signatures of fragmentation, and so you would see more genetic structure. But in these long-lived species, maybe not so much. But honestly, I don't know the answer to that is. So I'd have to look it up.
So another question from John Birmingham Jr. And I think this might have been somewhat of a joke of a question, but I'm actually very interested. Especially just some background information about myself, right? So I've studied monarch butterflies, and you always hear about milkweed and how everyone needs to plant milkweed. So this fantastic question by John is, should I burn the milkweed patch in my backyard? Because you've shown that could increase the ability of them to flower and increase fecundity.
Well, I guess so. The answer is you probably should not burn in your backyard unless you own a prairie and you have many acres. There might be a fire hazard. So certainly, I know that like prairie species are really strongly they react positively to fire. Generally, I don't think so I have milkweeds living in my yard as well. I honestly have never had a problem with fecundity in my yard milkweeds. I think because the population sizes are big enough of most of the milkweed species around here that you don't need to kind of intensively manage them to promote reproduction. So I would say no, don't burn them. But if you have a prairie, then you should burn your prairie. So if you if you do that, you should probably have people who are trained in fire to make sure that you don't set your house on fire. Yeah, but there are resources out there. So for example, MDC can help you burn. We also have actually quite a few trained staff at the Shaw Nature Reserve who are trained in fire and doing controlled burns. And they burn out there all the time. Yeah, in dormant season burns.
Excellent. Yeah, I bet the fire department's never been called for I was trying to control burn my milk. So,
my flower bed.
Yeah. I hope not. All right. I think Yusan will take the next question. Thanks for that. That was very interesting.
Mm-hmm.
All right. I actually have a question myself. So it was kind of prompted by your map of different genotypes or different genets. Is the spatial distribution within a site like important? Like, or is that like smaller in the scale of the pollination network?
So can you repeat that?
Oh, I'm just wondering, like, if the distribution of different genotypes within one site, like, say, if they're like clumped together or like distributed in different places, is that important at all? Like, considering the pollination.
So I think it certainly could be. Yes. So I think we looked at density, but the problem with some of the sites in our study is that, yeah, some of them were, spread out across like huge prairies, and some of them were kind of clustered within a tiny small prairie. Certainly, I think both like a population size, but also denser populations is probably going to facilitate. So if you have a clump of genetically different individuals and 50 flowering individuals at once, you're probably going to have greater pollination success than a site where the plants are spread out over thousands of acres. So I think there is probably a density effect, but unfortunately the sites that we chose weren't really great in order to test the density. So that would be something that we'd have to do in the future to really get a 100% clear answer on that. But my guess is that yes, density probably has an important component to pollination success.
Thank you.
All right, I think we got maybe time for one more quick question. So I'll go with Matt Austin's question. He says, fascinating work. I'm intrigued by the pollen limitation hypothesis to explain patterns of mead's milkweed fecundity. So how much is known about the viability of hybrids produced from crosses between mead's milkweed and sympatric milkweed species?
Oh, I don't know at all. I've never seen, yeah, because I think there's something like 17 milkweeds in Missouri. And, when we were out in the field, we would regularly see 4 or 5, co-occurring species of milkweeds. We didn't really see any evidence for hybridization. And so hybrid, conspecific pollen could be kind of a pollen interference that could be causing lower fecundity in small populations as well. So that could be another factor. I don't know of any studies on hybridization in milkweeds, but we didn't see any evidence in these ones that we worked on. So yeah, that certainly could be a factor limiting reproduction and kind of exacerbating the pollen limitation.
Perfect. Yeah, and he actually just put another thing in the chat. So I think you two definitely should talk. I'm sure you guys probably already talked, but
actually we haven't, because I think he started and it's always been in time of coronavirus. Actually, I would love for somebody else to do all these pollination biology experiments that I just proposed. So if anybody's interested, let me know, because I'm not really a pollination biologist, I'm a population geneticist.
I happen to know of one, and his name is Matt Austin.
Yes, okay, well, Matt, you need to come and talk to me and see if you're interested in working on pollination biology and Mead's milkweed. Yeah.
Well, thank you so much, Dr. Edwards. I'm sure your kids have had enough time at the park now and they want to come home.
They do, I'm sure. Yeah.
So we'll let you go. Thanks so much. And to all of you that are watching right now, we look forward to seeing you next week, same time, and on the YouTube channel. Thanks so much, Dr. Edwards, and hopefully we'll all be able to see each other soon in person.
Yes, thanks everybody for watching. I appreciate it.
All right, have a good night all.
All right, thank you.
All right, the stream is ended.